Okay, good evening, students. Today we're going to learn uh, few basic concepts of evolution. Okay, so coming to evolution, the term evolution is coined by Herbert Spencer. Okay, we know it is nothing but the evolution is a branch of science which deals with the gradual diversification of complex organisms from simple organisms in due course of time with respect to adaptation caused by a change in the environment. That is what actually evolution means. Here, there are four aspects involved in this concept. One is origin of complex organisms or evolution of complex organisms from simple organisms. Second one, why they have to evolve? Because they have to evolve because there is a need that has arisen for them to evolve. Why that particular need has arisen? Because there is a change in the environment and the animal or the organism has to adapt itself to the newly changed environment. That is the basic idea behind evolution. So no evolution will take place unless otherwise there is a need. What is this need? The need is a change in environment. So when the change in the environment takes place, the organism tries to adapt itself to the newly changed environment. As the organism tries to adapt to the newly changed environment, it brings about a change in its genetic composition, which results in genetic divergence. Genetic divergence that results in variations in phenotypes, so that organisms which are almost all are similar, now they are said to be morphologically dissimilar. In due course of time, if reproductive isolation sets in, then we say that evolution has taken place. So whatever that I have told you, you may not be able to understand all these aspects, isn't it? So let me clear this in a simple and better way. Before going to this, I would like to ask you one question. How do you define a species? Anyone can tell me.
Uh, Organisms which can reproduce within themselves అంటే వాటి సేమ్ అంటే వాటిలో వాటి రిప్రొడ్యూస్
Next, any other
Species is the basic unit of classification.
basic unit of classification? Excellent. Any other
Sir, a group of individuals.
group of individuals? Hmm.
Sir, a group of individuals which shows the similar characters.
A group of individuals which show similar characters. Very good. Next, Any
So
other?
they will be genetically identical.
Genetically identical
Um,
means only twins. Only twins are genetically identical. That means twins only belong to the same species. We do not.
yes, sir. Yes, sir.
That means you are not Homo sapiens.
Sir?
You are saying that they are genetically identical. Genetically identical means
Uh,
only identical twins are genetically identical.
yes,
Tanmay,
sir. Yes,
Dirija,
sir. Sorry.
Tanmay and Samuels are not genetically identical, but
Yes,
they
sir.
belong to the
Yes.
same species Homo sapiens.
Sir, I
Okay,
mean that they look similar.
they look similar. Similar in the sense you and me are similar.
Phenotypically,
Phenotypically, in what way?
they may be of the same gene pool.
I mean, we have the same gene pool. What, what does gene pool mean?
Uh,
Any other answers? Rishi, Lakshmi, Vasu.
రీప్రొడ్యూస్ చేయగలిగే కెపాసిటీ ఉండాలి సార్.
ఆల్రెడీ చెప్పారు. వినయ్ చెప్పాడు. వైష్ణవి, Safia Begum.
Sir, a group of a group of organisms create the natural for offspring, sir.
Very good. Which produce offspring. Then, if they are not able to produce offspring, can't we call them as Homo sapiens? There are some human beings which which are said to be, we cannot say that they are sterile, but still due to certain hormonal imbalances or something like that, they are not able to produce offsprings. You won't call them as Homo sapiens. So, what I want to insist on this aspect is you need to study thoroughly every aspect that you're going to take into consideration. You must have a clear cut idea if you want to get into central universities, or if you want to become a lecturer, you want to become a professor. The same question the interview board asks. They ask only fundamentals. They don't go for the what is starter box, what is Pribno box. They won't ask you. They just want to know whether you are familiar with the fundamentals or not. So be familiar with all the fundamental aspects of science. So let me explain this. Species. Species, as one of you said, are the individuals which are morphologically similar but not identical. If you want to write, you can write it down. They are morphologically similar but not identical. So they are not. All the individuals that belong to the same species need not be identical, but we have to be similar with respect to morphology. So when you see some individual, if you are going to call him as human being, that means you have some idea about the characteristics of human being, the morphological features of human being, isn't it? So those characteristics which are said to be seen in all the individuals that belong to the same species, we call those individuals and the characters as morphologically similar characters. For example, you see some animal, you call it as a dog, you call it as an elephant. Why? Because they are having certain common features. All the species may be different, the individual, the genera may be different, but here they are said to be having certain common features. But if you say they are morphologically similar, but different species of elephant, different species of dogs, different species of other animals come under also the same species. But this definition is not enough. So the second characteristic that is important for understanding species is they have the same chromosomal configuration. They have the same chromosomal configuration. For example, Homo sapiens. We belong to the sapiens species. And this sapiens species, we all have 46 chromosomes. Whether you live in India, whether you are Chinese, whether you are an American or you are an Egyptian, we all have 46 chromosomes only. That means we have the same chromosomal configuration. And that is not going to fit into the definition, these two characteristics. Another feature is there for the species. That is, they must interbreed among themselves. The individuals of a species must interbreed among themselves, and in so doing, they must produce a fertile offspring. Okay, that means when human being marries a human being, he must produce a human being only, and a human being it must be able to reproduce only with the human being. That is what interbreeding is. In a garden, there is a rose flowered plant, and by the side of which there is a jasmine flowered plant. Let's say both exhibit wind pollination. The pollen come and fall on the stigma of this plant. The pollen of jasmine plant come and fall on the stigma of this plant. Still too, there is no fertilization happening because though the pollen fall here, the pollen tube do not germinate because they belong to two different species. That means these two cannot interbreed. So interbreeding must be one of the important features of a species. And in so doing, as they interbreed, they have to produce offsprings, children, but they must be fertile. Why I am calling this so? A scientist wanted to know what will happen if, what is that? Donkey is crossed with a horse. And he found that they gave rise to mule. So interbreeding happened between these two individuals. Donkey belongs to Equus cabala species and horse belongs to Equus equa species. That means they two belong to two different species. Still, they are interbreeding. Can we call them as species because they are interbreeding? No, because they have to produce a fertile offspring. But here, the mule is found to be a sterile offspring. That is the reason why we say that these two are not belonging to the same species. So they must interbreed and produce a fertile offspring. That is another characteristic feature. And the next one is as they interbreed. For example, human population living in Delhi. Okay. Previously, about 1000 years back, around Delhi there is a dense forest, isn't it? So that the people of Delhi are not having any contact with the people present at the side. So whatever the interbreeding that takes place among these individuals only. Likewise, there is African continent, and we call this as dark continent because Nobody knows it exists. Whatever the individuals that are present there, they only can interbreed among themselves. Till Columbus discovered, we do not know America itself. So whatever the reproduction that has taken place, that is restricted to that particular area only. Let's say there are 1,000 individuals here, and each individual is a diploid human being. That means each chromosome has a number of loci, isn't it? So let's say there are 1,000 individuals for a particular locus. For example, eye color, we have 2,000 genes, isn't it? For a particular locus, because we are diploid, each character is determined by pair of genes, alleles, and thus we have 2,000 genes. And some of these are said to be black color, some of these are brown color, some of these are like Aishwarya Rai blue color, and some of these like Hrithik Roshan green color. So different colors are there. But these two constitute 90%, these two constitute only 10%. Even if so, all are said to be present in the same area. And with respect to eye color, when they interbreed, the same characters are said to be shared among the individuals of that population. And when you consider all the genes present on all the 46 or 23 pairs of chromosomes, they are also shared. So all the genes present on the total number of chromosomes, in short, haploid set of chromosomes, because whatever the genes that are present here, they are also present here. So we need not consider it as two. So all the genes present on different loci in a haploid set of chromosomes constitute gene pool. As the organisms belonging to the same species, as they interbreed, what they do, they share a common gene pool. Okay, this is what I wanted to say. If we living in Visakhapatnam are isolated from from the entire world, we don't have any connection, no vehicles, no transportation through land, no transportation through air, no transportation through even waterways. So what happens? We are completely isolated, and whatever the reproduction takes place, it is among the individuals that live in Visakhapatnam only. Then what happens? We share all the genes among ourselves only. That means all the gene pool is shared among all the individuals that belong to that particular population of a species. Thus, they share a common gene pool because they interbreed among themselves. Next one is they occupy an ecological niche. The individuals of a species occupy the same ecological niche. Let's see, there is a big forest. There is a big forest. In this forest, there is a lion, there is a tiger, there are several trees, large trees, and several plants, even grasses are present. And even small ants and other animals, insects are also present. And thus, the forest is the place where all these organisms, wild animals live, which is called as habitat. What is habitat? Habitat is the physical space occupied by an organism. So when somebody asks you, where is the lion? You say, lions are in forest. When somebody asks you, where is the tiger? You say, it is in forest. But actually, where in the forest the lion is present? Then you say it is present in a cave. So in the habitat, there is a small area where this individual lives. And the individual that is living here will not allow the individuals that belong to another species. That means here the individuals that belong to the same species only live. And this is the total environment. The forest is a total environment, and this is a small microenvironment. And that microenvironment is called as niche. So all the individuals that belong to the same species occupy the same ecological niche. So I don't want to go into the details of niche. There is more to teach you on this aspect, but it is not relevant to to our particular topic of evolution. And all these are fulfilled. Still, too, we cannot call these individuals as a same species, though they are morphologically similar, though they have the same chromosomal configuration, though they interbreed among themselves, producing fertile offspring and sharing a common gene pool and occupying the same ecological niche, we can't call them as the same species. And what they have to do, the last thing is, they must be reproductively reproductively isolated from other such groups. They must be reproductively isolated from other such groups. Then only we say that these individuals belong to one species. And evolution is nothing but formation of complex organisms from simple organisms, during which what happens? Species A gradually diversifies to form species B, and species B gradually diversifies to form species C. Thus, new species arise. Then only you say evolution has taken place. Thus, evolution simply means origin of new species. Without the origin of new species, you cannot say evolution has taken place. For the evolution to take place and origin of new species to take place, all these characteristics have to be fulfilled. We know there are several monkeys. Monkeys are morphologically similar, and some of the monkeys that belong to different species may also share the same chromosomal configuration. Species A has 20 chromosomes and species B also has 20 chromosomes. That doesn't mean that they are said to be belonging to the same species, and they exhibit interbreeding, but they fail to produce a fertile offspring, then we say that they belong to different species. If they produce a fertile offspring, then we say that they are not different species. They belong to the same species. So the only thing that I want to mention here is that they must be reproductively isolated from the individuals which belong to another species, so that there is no interbreeding at all. Even if interbreeding takes place, like the what has done between donkey and horse, you will come across with a sterile individual which distinguishes them as two different species. So this is what the general characteristics of species is. Now I want to ask you one more question. Did you hear anything about subspecies?
Yes, sir. In trinomial nomenclature, subspecies is present, sir. There are example of this is Carvus splendens splendens.
Excellent. So we come across with subspecies in trinomial
And
nomenclature.
humans also Homo sapiens sapiens, sir.
Homo sapiens sapiens. So there is Corvus,
Splendens, splendens.
Splendens,
Splendens
splendens.
splendens.
only one.
Naja, Naja, sir.
Uh-huh. In Corvus.
Sir, Naja Naja, it's a taxonomy, sir.
Taxonomy.
Yes, yes, yes. Sorry, sir.
Not asking about Naja Naja. Corvus splendens. This is generic name. This is specific name. This is subspecies name.
Subspecies.
So this species is having only one subspecies or more than one.
Sir, more than one, sir.
Okay. What are the
Carvus
names?
splendens insolens, one more
Very good.
species, subspecies, sir.
Corvus
And as
splendens.
Carvus splendens One more Sri Lankan and Myanmar.
Very good. One more is Proteozoa. Okay. So there are three subspecies in Corvus splendens. Okay. We are saying that species A gives rise in due course of time to B. Then how come these subspecies evolved? How you distinguish between a
Sorry, sir.
just now I have told you that origin of new species has to take place so that we say that evolution is happening, isn't it? From simple organism, complex organisms are evolved. That means what does it mean? New species are being formed. We know that species give rise to new species. Then how come species gives rise to subspecies?
Some morphological changes takes
Uh,
place.
uh,
How you define a subspecies? Species definition we also in already chapter. How you are going to define a subspecies and how they originate. If you know these two things, evolution, you know everything about evolution. Okay, let me explain. Previously, all the crows belonging to the species Corvus splendens are present in India only. Okay, Corvus splendens, crows belonging to the species Corvus splendens are present in India. And due to competition, some of them got migrated to Sri Lanka, some of them got migrated to Pakistan, and some of them got migrated to Myanmar, that is Burma, erstwhile Burma, Myanmar. Okay, now what happened? The environmental forces that are operating in India are different from those that are operating in Myanmar, Sri Lanka, and Pakistan. So the same crow went to different places. Here also the same Corvus splendens is there, Corvus splendens is there, Corvus splendens is there, same crow is present. But what happened here? Because they are separated from each other, they cannot come back again. They have gone to a new place. Now what happened? They have to adjust themselves to the newly changed environment in Myanmar, and some to the environment in Sri Lanka, and some to the environment in Pakistan. What happened?
Adaptation, sir.
They have to adapt to themselves. Very good, excellent. So adaptation should take place. How the adaptation takes place? By bringing about certain phenotypic changes so that there is a morphological dissimilarity in due course of time. Okay, and this morphological dissimilarity arises due to what we call change in the genotype. So when there is occurs a change in the gene only, there is only a change in the phenotype. So because phenotype is determined by genotype, so unless otherwise there is a change in genotype, phenotype will never change. So after 100 years, I have gone to Pakistan and have observed that the crow that is present here is somewhat different from the crow that is present here. And the crow that is present here in India is somewhat different from the one that is present in Myanmar. And even the ones that are present in Myanmar and Pakistan, and Myanmar and Sri Lanka, and Sri Lanka and India are different from each other. How I am distinguishing them? Based on certain morphological features. There is a Chinese individual. When you see a Chinese individual, you can say he is a Chinese. When you see an Indian anywhere in the world, you can easily identify an Indian. But there are certain morphological changes. Likewise, here also there are certain morphological changes in all these crows. And scientists said that this one, that one that lives in Myanmar, is a new species. And another one said that the one that living in Sri Lanka is also a new species. Whereas the ones that are present in India and Pakistan, they are almost the same. It's just very minor that but we cannot distinguish between. They are said to be the same species. So that based on morphological features, they are saying that there are three different species. Now you have to prove that the crow that is living in Myanmar is a new species. How will you prove it? Based on morphology, somebody said that it is a new species. Will you agree? Let's say it is having white feathers. The crow that is present in India is having black feathers. The one in Myanmar is having white feathers. Can you say it is a new species or not? Yes or no?
సార్ మళ్ళీ ఒకసారి అడగాను సార్.
ఇప్పుడు ఇండియాలో ఉన్న కాకి పాకిస్తాన్, బర్మా, శ్రీలంక వెళ్ళిపోయిందిరా. బర్మా వెళ్ళిపోయిన కాకి, శ్రీలంక వెళ్ళిపోయిన కాకులు వాటిని సైంటిస్ట్ వెళ్ళి చూస్తే ఇండియాలో ఉన్న కాకి కంటే పూర్తిగా భిన్నంగా కనబడింది. సో పూర్తిగా భిన్నంగా కనబడినప్పుడు ఎలాగా దాని ఫెదర్ కలర్ తీసుకున్నాడు. ఇండియాలో ఉన్న కాకి నల్ల ఫెదర్స్ ఉన్నాయి, మయన్మార్లో కాకి గ్రే ఫెదర్స్ ఉన్నాయి, శ్రీలంకలో ఉన్న కాకి వైట్ ఫెదర్స్ ఉన్నాయి. ఇప్పుడు చూడగానే సైంటిస్ట్ ఏమనుకుంటాడు, ఈ మూడు స్పీసీస్ అని అనుకుంటాడు. అనుకొని పుస్తకం కూడా రాసేసాడు. ఇప్పుడు నీ దగ్గర తీసుకొచ్చి నీకు చూపించు, నువ్వు సీనియర్ సైంటిస్ట్, నేను కొత్త స్పీసీస్ రెండు ఐడెంటిఫై చేశాను అని అంటే నువ్వు మయన్మార్లో ఉన్నటువంటి గ్రే ఫెదర్స్ ఉన్న కాకిని, శ్రీలంకలో ఉన్న వైట్ ఫెదర్స్ ఉన్న కాకిని చూసి కొత్త స్పీసీస్ అని ఒప్పుకుంటావా?
ఫస్ట్ దాని క్యారెక్టర్స్ ని అబ్జర్వ్ చేస్తాం కదా.
చేసేదాన్ని చేసి పట్టుకొచ్చాడు. మార్ఫలాజికల్ క్యారెక్టర్స్ చాలా తేడా వచ్చేసాయి, ఒక 5-6 క్యారెక్టర్స్ తేడా వచ్చేసాయి. ఇప్పుడు ఏం చెప్తావు, సేమ్ అంటావా?
వద్దు సార్.
సార్ ఇట్ ఈస్ నాట్ పాసిబుల్ సార్. ఎందుకంటే మనం స్పీసీస్ మొత్తానికి ఒక ఐ మీన్ ఒక దగ్గర స్టోర్ చేసుకుంటాం కదా సార్ స్పీసీస్ మొత్తాన్ని. అందులో ఈ క్యారెక్టర్స్ ని సిమిలర్ గా మొత్తం థరోలీ చెక్ చేసిన తర్వాతే కదా సార్ మనం న్యూ స్పీసీస్ అని చెప్పేసి వాటికి నేమ్ ఇవ్వడం జరుగుతుంది.
నువ్వైతే ఏం చేస్తావు?
According to ICZF, they have some characters, sir, about Carva splendens. So they identify it's not a new species, it's a subspecies of a particular species.
అది ఎలా చెప్తావు? నీకు దగ్గర బుక్ కూడా లేదు. యూ డోంట్ హ్యావ్ ఎనీ ICZF రూల్ బుక్. దెన్ కాంట్ యూ సే దట్ it is not they do not belong to the same species, or they belong to the
ఫెర్టైల్ ఇండివిజువల్ వస్తే అప్పుడు దాన్ని.
Very good. What you have to do immediately is the individuals that belong to the same species intervene among themselves, isn't it? So here you have to see, you bring the crow from Myanmar and try to cross these two, mate these two. And if interbreeding takes place and fertile offspring is produced, then you can say that they are not two different species because they are interbreeding and they are producing a fertile offspring. And the same thing you do with the Sri Lankan also. Then also they are said to be interbreeding and producing fertile offspring. When they produce a fertile offspring, you cannot say that they are two different species. So I have put forward certain characteristics with respect to species. They must be morphologically similar, but here these are morphologically dissimilar. They must they must have the same chromosomal configuration. So here they are having the same chromosomal configuration. They must interbreed themselves. So they are interbreeding them among themselves. They must produce a fertile offspring. They are producing a fertile offspring. They must share a common gene pool because they are living in Myanmar. They are sharing a common gene pool, and they have a common ecological niche, and they are not reproductively isolated. They can interbreed, so that they are not reproductively isolated. When they are not reproductively isolated, we say that they belong to the same species. But same species must be morphologically similar. But these are morphologically dissimilar. So how you define a subspecies is a subspecies is individuals of a species. These are nothing but individuals of a species which are geographically isolated, which are geographically isolated because they are said to be present in different places. Morphologically dissimilar, morphologically dissimilar, but are not reproductively isolated. So when there is no reproductive isolation, there is no formation of a species at all. That means species evolved up to subspecies only. Let's say it is subspecies A. And if it is exhibiting reproductive isolation, then it will form into a new species. So in evolution, it has reached only up to the subspecies level. Here, evolutionary process is going on and a new species to be formed, reproductive isolation should take place. Here there is no reproductive isolation because they are interbreeding among themselves. That means it is stopped in the between the formation of a new species from an old species. And this is what we call it as subspecies. How a subspecies arises? When the individuals of a species are geographically isolated. This is the crux of evolution. So, the crux of evolution is they must be geographically isolated and they must exhibit reproductive isolation. Just if the geographical isolation is not sufficient, they must exhibit reproductive isolation too. So when these two things happen, you say there is happening a process of evolution where a new species is originated. Okay, and what actually brings about reproductive isolation? So before go into that, I want to have a third question. Donkey married to horse gave rise to mule, which you call it as sterile. Okay, what makes mule sterile? That's my question. What makes mule sterile? Who is going to tell?
It is a non-identical.
క్రోమోజోమ్స్ డిఫరెన్స్ ఉంటాయి కదా సార్. హార్స్ క్రోమోజోమ్స్ డాంకీ క్రోమోజోమ్స్ డిఫరెన్స్ ఉంటాయి కదా సార్. ఒకవేళ సేమ్ క్రోమోజోమ్స్ మ్యాచ్ అవ్వదు కదా, అంటే ఏదో ఒకటి ఎక్కువైనా తక్కువైపోయినా ఉండిపోతుంది కదా సార్. సో అవి
హార్స్లో 20 ఉన్నాయి, డాంకీలో కూడా 20 ఉన్నాయి. అప్పుడు number of chromosomes in donkey and horse are same. So it receives 10 from donkey, 10 from horse. Still you are saying it is mule.
గేమ్స్ ఏమి ఉండవేమో కదా సార్, అంటే అక్కడ ఉన్న పర్టికులర్ ప్లేస్ లో ఉండవేమో కదా సార్.
లేకపోతే మరి
Sir, it is a sterile offspring.
it is a sterile offspring. That's what I'm asking. What makes mule sterile? Why are you saying that it is sterile?
Because of genetical changes, sir.
Genetical
Of
change.
course, they both are different species.
అసలు స్టెరైల్ అని ఎందుకు అంటావు? ఎప్పుడు అంటావు? When you declare an animal or a plant sterile,
Infertile, sir.
means
Not
They are not fertile.
Yes, sir.
You're saying an antonym for sterile.
Sir, they have no able to produce offspring, sir. Sterile means fertile means they produce offspring.
So they are not able to produce offsprings. Why they are not able to produce
Offsprings
offspring?
that is called sterile offspring.
They are not able to produce offsprings. When that will happen? When mule is married to either horse or donkey or another mule only, that will happen, isn't it? But you're not getting it crossed with any other thing. Still, too, you can say that it is sterile. How? దేంతో క్రాస్ చేసి పిల్లలు పుట్టకపోతే నువ్వు స్టెరైల్ అంటావు కదా, కానీ క్రాస్ చేయకపోయినా దాన్ని స్టెరైల్ అని చెప్పగలవా?
లైక్ క్రోమోజోమ్స్ అది ఒకవేళ మియోసిస్ అయినప్పుడు డివైడ్ అవ్వదు కదా సార్ అది.
కాక ఎందుకు మామూలుగా చెప్పు, డోంట్ గో టు ద క్రోమోసోమ్ నా జీన్ లెవెల్. స్టెరైల్ అని ఎప్పుడు అంటావు ఒక యానిమల్ని?
So the secondary sexual characters are not developed, sir.
సెకండరీ సెక్షువల్ క్యారెక్టర్స్. ఇంకా దానికి సంబంధం ఏమైనా ఉందా? గడ్డం లేని మగాళ్ళు ఉంటారు, అప్పుడు కొంతమంది గడ్డమే రాదు, సెకండరీ సెక్షువల్ క్యారెక్టర్స్ లేవు, వాళ్ళు స్టెరైల్ అని ఇస్తామా? హ్యూమన్ బీయింగ్స్లోనే ఒక ఇండివిజువల్ని స్టెరైల్ అని నువ్వు ఎలాగంటావు చెప్పుకొని? సరే, ఫెర్టైల్ అన్నావు కదా, ఫెర్టైల్ అన్న ఇండివిజువల్కి ఒక క్యారెక్టరిస్టిక్ చెప్పు. వాట్ మేక్స్ అన్ ఇండివిజువల్ ఫెర్టైల్? ఒక ఆర్గానిజం ఫెర్టైల్ అని ఎప్పుడు అంటావు?
Produce the offspring.
ఆఫ్స్ప్రింగ్ ప్రొడ్యూస్ చేయాలి. ఆఫ్స్ప్రింగ్ ప్రొడ్యూస్ చేయాలంటే రిప్రొడక్షన్ జరగాలి కదా. రిప్రొడక్షన్ జరగాలంటే ఏం కావాలి?
Gametes.
Fertilization, sir, to production of gametes.
Production of gametes should take place. That is what the answer is. So, if the organism is not able to produce gametes, if the organism is not able to produce gametes, then it is sterile automatically. Without gametes, there is no reproduction, there is no fertilization, there is no offspring at all. So, మీరు offsprings దాకా వెళ్ళక్కర్లా, అది గ్యామెట్స్ ప్రొడ్యూస్ చేస్తా లేదో చూస్తే చాలు, గ్యామెట్స్ ప్రొడ్యూస్ చేయలేదు. So, it is unable to produce gametes. So, why mule is unable to produce gametes? So, for that you have to tell me how gametes are produced. What is the process that produces the gametes?
Spermatogenesis and oogenesis, sir. Gametogenesis.
What happens during gametogenesis? What happens during
The
gametogenesis?
production of
Number is reduced to half, sir.
How? What is the process is called as? That process is called as?
meiosis.
So, in order to produce gametes, meiosis should happen. In meiosis, there is prophase 1, metaphase 1, anaphase 1, telophase 1. Likewise, prophase 2, metaphase 2, anaphase 2, telophase 2. In prophase 1, in zygote, what happens?
Homologous chromosomes comes to
Synapses.
synapses form అయ్యి దగ్గరకు వస్తాయి.
Pairing of homologous chromosomes takes place. So, what do you mean by homologous chromosome?
Same chromosome.
Same chromosome. How it becomes the same chromosome? First chromosome, there is a second chromosome. Can you call these two as homologous? No. Why?
లాస్ట్ సైజ్ జీన్స్ సేమ్ ఒక్క దగ్గర ఉంటాయి సార్ దీనికి వాటికి.
So the loss is
Based on the genes and the presence of the centromeres, sir.
వెరీ గుడ్. పెయిరింగ్ ఎలా జరుగుతుంది అని అంటే, ఫస్ట్ క్రోమోజోమ్ ఇక్కడ ఎక్కడో ఉంది న్యూక్లియస్ లో, దాని కౌంటర్ పార్ట్ ఇంకెక్కడో ఉంది. ఈ రెండు దగ్గరికి వచ్చినప్పుడు దాంతోపాటు రెండవది కూడా అక్కడికే వస్తుంది, కానీ ఈ రెండింటికి మధ్యలో పేరింగ్ జరుగుతుంది. ఈ ఒకటో దాని దగ్గరికి ఇది వచ్చినప్పుడు మాత్రమే పేరింగ్ జరుగుతుంది. ఎందుకట? ఫస్ట్ ఆఫ్ ఆల్ both are having the same centromeric position. Both are metacentric chromosomes or submetacentric chromosomes likewise. First thing. Second thing that makes them pair is their length is same. Their length is same. So lengthwise, the pairing is lengthwise. The pairing is based on centromeric position. Third one is the loci are same. The first locus is for skin color, first locus on that counterpart, this chromosome is also for skin color. Second locus for eye color, second locus on this counterpart is also for eye color. That means all the loci on these two chromosomes are similar. But here in case of when the horse is crossed to a donkey, one, as when I said, if this is having 20 chromosomes and this is having 18 chromosomes, it produces gametes with 10 each, and this produces gametes with 9 each. And when they combine, this mule is produced, it is having 19 chromosomes, where there are 9 pairs and one is left. And it is going for meiosis. The reproductive is also going for meiosis. In meiosis, pairing takes place. 9 chromosomes pair up and one chromosome is left outside. So what happens? Meiosis will not happen. There is no gametogenesis at all, and this becomes a sterile individual. Let's say even if there are 20 chromosomes and they are sharing 10 each, so it is having 10, but these 10 are not 10 pairs, because the first chromosome of horse may be long and the first chromosome of donkey may be somewhat short. It is metacentric and it is submetacentric, and pairing will not take place. So even if the number of chromosomes is same, there is no chance for the pairing to take place. So when the individual is unable to produce gamete due to abnormal meiosis, what happens? We call it as a sterile individual. Abnormal meiosis, why it takes place? Because there is a change in the, even if they are same, the chromosomal configuration is somewhat different. So there is a change in the chromosomal configuration. So when there is a chromosomal change in the chromosomal configuration, what happens? There is reproductive isolation. So even donkey is crossed with horse and producing mule, we cannot say that they are interbreeding among themselves. If they interbreed among themselves, they must produce a fertile offspring. They are interbreeding, but they are not producing fertile offsprings, and then we say that they are reproductively isolated. So reproductive isolation can happen only when there is a change in the chromosomal configuration. For example, chimpanzees. Scientists say human beings are originated from chimpanzees. Chimpanzees have 48 chromosomes and human beings have 46 chromosomes. While I was explaining you the structural variations in chromosomes, I have explained you about translocation mutation. And one such translocation mutation is Robertsonian mutation. In Robertsonian translocation mutation, what happened? Two chromosomes, one gets cut here, and another one is also acrocentric. It gets cut here, and this one is translocated onto this one. What happens now? In place of two chromosomes, there is only one chromosome. Thus, if two translocations such take place, instead of 48, the resulting individual will be having 46 chromosomes. So both are there in the same place. After this process, both are present in the same place. So let's say, like donkey and horse, they have reproduced, and it produces 24 chromosomes in the gamete, and this produced 23 in the gamete. Got fertilized, and the individual is have 47. So a new individual is produced. Now this new individual must be a fertile individual, and fertile individual must be able to produce gametes. But this is not able to produce gametes because 23 may pair and one will be left out, and the 23 may not pair even, because there is a change in the chromosomal configuration. Then you say that these individuals, when they keep on reproducing and they keep on producing only sterile offsprings, sometime after they decide that we are not compatible, and that leaves the one species leaves that space, and it will evolve differently. And in due course of time, so many genetic modifications, gene mutations occur, and morphological changes occur, and in due course of time, maybe 1,000 years, 2,000 years, or 10,000 years, we cannot see any similarity between these two. One thing, they are morphologically dissimilar now, and even their chromosomal configuration is not same. Then we say that a new species is evolved. So a new species to evolve, what is the first criterion? Reproductive isolation should take place. How the reproductive isolation takes place? By a change in chromosomal configuration. How a change in chromosomal configuration occurs? Only by mutations. That is the reason why scientists call mutations as raw materials for evolution. So for evolution to happen, there must be mutations. Mutations are raw materials for evolution, where they bring about a change in the chromosomal configuration, which results in reproductive isolation, so that a new species is formed. In due course of mutations, if the mutations are chromosomal mutations, they bring about a change in the chromosomal configuration. No problem. There is a Robertsonian translocation. It is a chromosomal mutation. 48 to 46. There is a change in number of chromosomes. If it is a gene mutation, what happens? There is only small changes in the genotype, so that it results a change in the phenotype. That means there is a morphological dissimilarity, which results in the formation of subspecies only. So from species A, what happened? Only gene mutations occurred. It resulted in subspecies. When chromosomal mutations take place, and when there is a chromosomal configuration, when reproductive isolation takes place, this subspecies eventually will evolve into a new species. That is what actually the crux of evolution is. And there is a scientist called as Hardy Weinberg. He proposed a law, Hardy-Weinberg law, which explains that not only mutations, there are other certain factors which also are responsible for evolution. Okay, and he makes his theory or law in this way. So he says that there is a population, and according to him, this population is a large population. There is a large population, and this large population is having individuals which are sexually reproducing individuals, and this sexually reproducing individual population is called as panmictic population, because this process is called as panmixis. So if they exhibit panmixis, they are sexually reproducing, and they during reproduction they exhibit random mating. So there are three criteria for Hardy-Weinberg law. The population should be large, and the reproduction should be sexual, and the mating should be random. So when these three conditions are there, what happens is that he suggests that the gene frequency remains constant. Gene frequency remains constant from one generation to another generation. From one generation to another generation. In the absence of selection, mutation, and gene flow. This is what his law is. So by putting it all these 7 points together, in a sexually reproducing, randomly mating, panmictic population which is large, gene frequency remains constant from one generation to another generation in the absence of selection, mutation, and gene flow. So, according to him, there are three criteria which bring about evolution. Okay, and he says that gene frequency remains constant from one generation to another generation. We are saying that Corvus splendens is there. It evolved to form Corvus splendens splendens. It evolved to form Corvus splendens insolens. It evolved to form Corvus splendens protegatus. What is happening here? There is a change in the genotype which brings about a change in the phenotype. When there is a change in the genotype, for example, there are red colored individuals, and all are said to be red eyed. For example, Drosophila. Some of these are white eyed individuals. So, previously there are 100 individuals which are red in color. All are red, all Drosophila are red eyed only. So, what is the frequency of this particular gene which brings about red color? It is 100%, isn't it? All the individuals. When what happened here? There is a mutation occurred. The gene for red got mutated to produce small r. Now this happened in about 10 individuals. Now previously there are 100 individuals which are red. Now there are only 90 individuals which are red, and there are 10 added which are said to be white. What happened? Due to mutation, during the course of evolution, due to mutation, the frequency of red individuals gone down from 100 to 90 because there is a mutation being occurred here. So the gene frequency of capital R is reduced. It changed from one generation where all are red eyed to another generation where 90 are red eyed, 10 are said to be white eyed. Isn't it? So this is what a change in the gene frequency. When there is a change in the gene frequency, in due course of time, a subspecies may be formed. In due course of time, if reproductive isolation sets in, a new species may be formed. That means here, when the mutations occur, when there is a change in the gene frequency occurs, there is every chance for evolution to occur. And here he is saying gene frequency remains constant. That means he suggests a hypothetical situation where there is no evolution at all. In parents, red and white are said to be 50-50. In next generation also red and white are said to be 50-50. In the next generation also red and white are 50-50. There is no change at all. Then when there is no change, do you say evolution is taking place? No. So here he is saying the same thing. The gene frequency is not changing, so that there is no evolution. So for evolution not to occur, what are the criteria? The population should be large, the mating should be random, and these three should be absent. Selection should not be there, mutation should not be there, and gene flow should not be there. In other words, reverse it. When selection is there, gene frequency changes. When mutation is there, like we have seen just in this example, gene frequency changes. When gene flow is there, gene frequency changes. That means evolution takes place. How evolution takes place? When there is a change in the gene frequency with respect to selection. Let us say there are red-eyed individual and white-eyed individual. These are 90 and these are 10. If the red-eyed individual get married to only red-eyed individuals, their proportion gradually increases. When the white-eyed individuals are not mating with the red-eyed individuals, their proportion gradually decreases. So that means when there is a selection, there is a change in the gene frequency. These increase to 220 and these remain just 20 only. 240. Previously 90 by 100 is the gene frequency of red, and now it is 220 by 240. That means the gene frequency somewhat got changed because of a process called as selection. So when selection is there, gene frequency changes. Likewise, there are 100 individuals which are red-eyed again. The same example I am telling you. Out of these, 10 individuals have gone. What happens? 80 are red-eyed, all are red-eyed, there is no change in the gene frequency at all. Let us say 80 are red-eyed among these 100 individuals, 20 are white-eyed. Okay, now what happened? There is a frequency 80 by 100 of red, 20 by 100 of white. Now, these white individuals, of these, about 5 white individuals have gone to another place. Now, how many are there? 95. Out of which 80 by 95 is red and 15 by 100 is said to be white. So, you can clearly see that there is a change in the gene frequency brought about in the white-eyed population. And here in the other place, they are said to be 5 by 5, 100%. The gene frequency is said to be increased. So here, organisms are migrating. As organisms are going out, they also carry the genes along with them. That means there is a gene flow. When you say migration, Samuel sir is going to some place. When he is going, his chromosomes are also going. All the characters, the character for his eye color, character for his skin color, characters for his height, character genes for his intellectuality, all he is taking away along with him. That means there is a gene flow along with the individuals. When there is a gene flow, there is a change in the gene frequency. That means three conditions are there: selection, mutation, and gene flow. These are the three processes which bring about evolution, according to Hardy-Weinberg law. If selection is absent, mutation is absent, gene flow is absent, there is no evolution at all. Let me explain the same thing with a small example. So there is a population of 100 individuals, of which we are concerned with only one locus. Let's say this is first locus and there is a genotype A and small a are there. So they can exist capital A small a, and small a small a. Three genotypes may exist, of which of these 100, 36 are capital A type, homozygous dominant genes, 36%, and of the heterozygous dominant, they are said to be 48, 36, 48, 70, 84, and the remaining are 16% this. So this is the normal population. Okay. So here this is the first generation, parental generation. Now what happened? Let's see the number of genes. So every individual has two loci for a particular gene, isn't it? So here, how many genes will be there? There are nearly 200 genes, isn't it? Now, this individual, these 36% individuals are said to be fertile and they produce gametes. How many gametes they produce? Among all these 100 individuals, 36% of the gametes belong to capital A. So coming to the gametes, 36% belong to capital A. That means all gametes are A type only. And coming to the small a, all gametes belong to small a only. And coming to this, 24% belong to capital A and 24% belong to small a. Okay. So thus, the number of gametes with capital A are 36 plus 24, that is 60%, and the number of gametes with small a are 24 plus 16, that is equal to 40%. Okay. So this is a large population. 100 individuals means that is a large population. According to Hardy-Weinberg, the population should be large. Okay. Now in this large population, 60% of the gametes are producing gametes. 60% of individuals are producing gametes with A gene, dominant A gene, and the 40% of gametes that are produced by these individuals are having small a gene. Okay. Now, second criteria is, this is a large population. The second criteria is sexually reproducing. That means they must reproduce sexually. So they have produced gametes means they are sexually reproducing. The third criteria is mating should be random. There must be no selection. That means Only these two never should mate, or these two should never mate. Mating should be among all these individuals. So let's say there is a random mating. So random mating, how it takes place? A sperm with capital A should fertilize a ovum with capital A. A sperm with capital A should fertilize a ovum with small a. In other words, a sperm with small a should fertilize a ovum with capital A, and in another case, a sperm with small a should fertilize a sperm with small a. So, what is the percentage of gamete produced? Capital A 60 and small a 40. So, there is 60 into 60. How many? 3600. By means 36%. 60 into 40. 2400. 100%. 24%. 40 into 60. That is 2400. 24%. 60. 40 into 40. 1600. That is 16%. What is this 60 and 60 are? Capital A genotype. What is this 24%? Capital A, small a. Another 24%, capital A, small a. Total 48%. And small a, 16%. So 36%. Capital A are children. And the children with 48% are said to be having capital A, small a. And the children which are 16% are having small a, small a. So this is the F1 generation. What happened here? In the parental generation, capital A, dominant homozygous are 36%. Even in the children, the same. The heterozygous are 48%. Even in the children, the same. Sorry. The homozygous recessive are 16%. Even in the children, the same. That means there is no change in the individuals. There is no change in their gene frequencies of capital A and small a. The same is said to be present. In that case, there is no evolution at all. Let us see what happens here. The first criterion is selection. Okay. So, if selection takes place between these two individuals, then what happens? They mate with these kind of gametes only. Maybe maximum this type. So, the gene frequency of A increases, and these are not allowed to mate with them because they are having somewhat different characteristic. So they only mate among themselves. 16% individuals mate among themselves means that population will not grow that much, whereas 78%, sorry, 84% of these individuals mating means here there are dominant genes capital A, capital A, capital A. Three are there. Only small recessive gene is there. That means when compared with this recessive gene, the dominant genes are more in number and their frequency changes when there is selection. Likewise, what happens? Mutation takes place. In small a 16% individuals, some of these, let us say 8% of individuals where there is mutation taking place and it got changed to capital A. What happens now? The frequency of small a decreases and the frequency of capital A increases. So when mutation is there, there is a change in the gene frequency. And the third criterion is gene flow. Out of this 48%, out of this 36%, let us say 20% have gone out. What happened now? 20% individuals capital A carried along with them capital A dominant genes along with them. So the gene frequency of capital A dominant gene decreases and the gene frequency of small a comparatively increases. So when there is a selection, mutation and gene flow, there is no change. There is a change in the gene frequency which brings about evolution. So thus, according to Hardy-Weinberg law, for evolution to take place, there must be always a large population. What happens if it is a small population? In a small population, if there is one change occurs, it will be immediately spreading to all the other individuals. For example, there is only population of 50 individuals. Of these, 10 individuals got mutated to produce red colored eyes. So when they mate among themselves, in due course of time, all the population may have red eyes because they are small enough. Let us say there is 1 lakh population. In 1 lakh population, only 10 got mutated to produce red eyes. Can, in due course of time, these 10 individuals, after reproducing with other individuals, can it make all 1 lakh individuals to be red eyed? No, not possible because it is a large population. So when there is a large population, there is no evolution. When there is random mating, there is no evolution at all. When there is selection not, mutation absent, and gene flow absent, there is no evolution at all. So here these three must operate to bring about evolution. So along with mutations, if there is selection, what Darwin said, natural selection. If there is gene flow, what the other scientists say, migration, and these three are considered as evolutionary forces with which evolution may take place. Thus, species A, in due course of time, if gene mutations take place, it produces only subspecies. We call this as microevolution. Evolution up to the formation of subspecies is called as microevolution. If chromosomal mutations take place or chromosomal configuration changes and reproductive isolation takes place, new species is formed, then we call it as macroevolution. And for microevolution or macroevolution to take place, there must be what we call a geographical isolation to operate here. When individuals separated, then only evolution starts. If all are present in the same place, all are said to be prone to the same ecological or environmental features, mutation will also be same, and we are interbreeding randomly so that all the characteristics are said to be passing on to the next generations. There is no change in the gene frequency. So there must be a geographical isolation that brings about the formation of a subspecies. If reproductive isolation sets in, there is evolution of new species. So this is what actually I want to tell you today about the concept of evolution. Okay, is it clear for everyone? Do you have any doubts?
Yes, sir.
So what is the primary requisite for evolution for the formation of a new species? కొత్త స్పీసీస్ రావాలంటే ఏం జరగాలి?
Mutations.
Reproductive isolation.
Only one thing, reproductive isolation should happen.
Yes, sir.
రిప్రొడక్టివ్ ఐసోలేషన్ జరిగితే కొత్త స్పీసీస్ వస్తుంది. ఆ రిప్రొడక్టివ్ ఐసోలేషన్ జరగాలంటే ఏం జరగాలి? మీకు మ్యూటేషన్స్ జరగాలి.
Gene mutation.
కాకుండా జీన్ మ్యూటేషన్ జరిగితే రిప్రొడక్టివ్ ఐసోలేషన్ రాదు.
Mutations, selections, gene flow. Yes.
Chromosomal mutations.
క్రోమోజోమల్ మ్యూటేషన్స్ జరిగితేనే, క్రోమోజోమల్ కాన్ఫిగరేషన్ లో చేంజ్ వస్తేనే, నెంబర్ ఆఫ్ క్రోమోజోమ్స్ చేంజ్ అయితేనే నీ కొత్త స్పీసీస్. ఫర్ ఎగ్జాంపుల్, there is no geographical isolation. There is no geographical isolation. Still to mutation happens by a mutation. Still to evolution happens by a mutation called as polyploidy. So what is polyploidy? There is a Gossypium herbaceum. This is cotton. Okay, its configuration is 2n. It has to produce haploid gametes, but there is a phenomenon called as non-disjunction. So due to non-disjunction, it produced diploid gametes. So there is another Gossypium herbaceum species, individual of the same species. It is 2n. It produced haploid gametes. Now this diploid gamete fertilized this haploid gamete, resulting in a triploid individual. So this triploid individual is morphologically dissimilar because it's having large capsules, it is having large leaves, large tree. So everything is said to be somewhat completely different with respect to morphological features. So we can say that this is a new species, but we cannot say simply like that. We have to mate this with this one. So 2n and 3n we tried, but 3n is not able to produce gametes because pairing will not take place between three chromosomes. It will take place only between homologous pairs of chromosomes. Then we say it is a sterile individual. When that is produced, we can say that a new species originated. So even in what happens, even if geographical isolation is absent, all the individuals are present in the same locality, then too also due to polyploidy, new species may arise. So what I want to explain you is here Gossipia herbaceae, which is 2n, is not able to reproduce with 3n because it is sterile. So what happened here? There is reproductive isolation happening here. So without reproductive isolation, new species will be formed. Without reproductive isolation, you can form only up to subspecies. How? Due to gene mutations which bring about morphological changes. Only when chromosomal mutations take place, then reproductive isolation takes place and a new species is formed. Okay. Even in case of human beings, the population is said to be large, isn't it? You say that we are said to be exhibiting selection and there is no random mating because we see jatakas, isn't it? And we select uh, a girl or a boy based on certain characteristic features. So I want a girl who is said to be somewhat beautiful with what we call with respect to her color complexion only I am seeing. So if that is the case, if all male are said to be selecting the female with respect to color complexion, or all female selecting with respect to male with respect to color complexion, what happens? There is a selective selection force taking place, and the gene frequency of those that are white will increase, those that are black will decrease. But that is not the case. Even in the human population, the mating is not having any selection at all. It is always random. పోనీ అలా కాదు, తెల్లగా ఉన్న వాళ్ళందరూ తెల్లగా ఉన్న వాళ్ళని చేసుకుంటున్నారు, నల్లగా ఉన్న వాళ్ళని ఇష్టపడట్లేదు అనుకుందాం. Still to we say that it is mating is right to be random. For example, there is a tongue, there is a feature in me that is called as tongue rolling. I can roll my tongue. I can roll my tongue. Many of you cannot do this because tongue rolling is said to be a dominant gene and non-rollers are said to be having recessive gene. Okay, so when I when I see a woman, will I be asking you whether you have a tongue rolling dominant gene or not? No, we don't ask at all, isn't it? Because they are said to be, for us, they are minor features. So with respect to tongue rolling, the mating is said to be random. There is a chemical called as phenylthiocarbamide, PTC. If someone places PTC on your tongue, you say it is somewhat sour or it's somewhat sweet. That means you can taste it, and they are called as tasters. And if you place the PTC on some other person's tongue, they cannot taste it at all. They cannot tell the taste. Then they are called as non-tasters. Okay. పెళ్లి చూపుకి వెళ్ళినప్పుడు నువ్వు ఈ పీటీసీ పట్టుకెళ్ళి అమ్మ నువ్వు టేస్టరో నాన్ టేస్టరో సెలెక్ట్ చేసి తెలుసుకుంటాను ఒక్కసారి నీ నాలుగు బయట పెట్టని నాలుగు మీద ఎవరైనా పెడతారా పెట్టడు ఎందుకోసం ఇలాంటివి ఉన్నాయనే తెలియదు సో దెర్ ఆర్ thousands of such characters which are not selected in human beings at all and thus mating is always random so selection జరిగిపోతుంది అనుకోవడానికి కూడా లేదు సో ఈ కండిషన్స్ బేస్ చేసుకొని ఎవల్యూషనరీ ప్రాసెస్ అనేది జరుగుతుంది ఓకే ఎనీ డౌట్స్ అప్ టు దిస్ పాయింట్ ఏమ్మా ఇప్పుడు చెప్పింది అర్థమైందా
Yes, sir.